I've tried to prepare a few comments about evidence for evolution and have done my best to keep it pretty non-technical. If there are aspects of the talk that you'd like a little more information about, I probably could direct you to some of the primary sources that would give you more of the basic information. But I think there's a lot to talk about with respect to what's going on in the field of evolutionary biology right now. My interest in evolution is probably derived from the fact that my basic training in science is in immunology, and one of my fundamental interests is between the interaction between hosts and pathogens. So I'm very interested in terms of how various pathogens interact with their host species. I'm very interested in sort of the evolutionary arms race that goes on. We're seeing a phenomenal example of that right now when we look at the human immunodeficiency virus and the sorts of genetic changes that it's going through as it is spreading through a wide swath of the human population. So that was my initial interest in biology, and then subsequently I've had the opportunity with a few of the different professors in philosophy at Chico State to teach a class called Science and Human Values. And that has been a very interesting area of reading and research and thinking for me over the last 10 years. And so I find that a very interesting way to access evolutionary biology, is to think about the impact that evolution has had on human development and our assemblage of various values. So this is, if any of you have seen the new movie Creation, one of the pictures, and any of you who have visited zoos or other primate centers know that there's quite an amazing moment when you go face to face with a non-human primate, and people realize the specialness of what happens in those sorts of encounters. So what are some of the new pieces of evidence? Well, a lot of times when we think about evolution, we go immediately to the fossil record. And indeed, Darwin was very concerned about the fact that the fossil record in his time was really quite scant, and he realized that there were very few transitional fossils, that if he's trying to show common descent, then the need to have more fossils was something that really plagued him, and it was one of the concerns, the biggest concerns that he had about defending his theory. Well, the good news is that we have an enormous number of transitionary fossils right now. For example, we have a very good idea in terms of how whales evolved. Some remarkable work and exciting things you can read about that. We also have a very good idea fairly recently in terms of the first animals that moved onto land. So what were the early tetrapods like? So we have, again, evidence like for that sort of thing. I'm not going to talk about that today, but I will bring up also the fact that we have an interesting discovery with respect to human ancestry, and that's an Ardipithecus species. This is interesting to me, particularly because what it says is that we've been thinking in sort of a wrong-headed fashion for many years in terms of what our common ancestor with chimps must have been like. We look at the chimps and we think, well, our common ancestor was very chimp-like. And here we go with Ardipithecus that's getting back in time very close to that common ancestor. And what do we find out? Ardipithecus, and this was a complete fossil, skeletal fossil that was found, and not too far from where some of the Australopithecine fossils were found, in Africa, of course. And what we find in this 110-pound woman, that she was basically a biped, a facultative biped, lived in trees, but also walked quite well. And for all intents and purposes, if you would make a comparison with Artie and modern-day women and female chimps, Artie looks more like the modern-day women. So our idea that somehow our ancestors were more chimp-like turns out probably to be a wrong-headed notion. And the fact is that we had these human-like features going back into probably greater than 4 million years ago. So things like that I find pretty exciting. As many of you may know, there's an amazing lack of fossils over on the chimp side, and the reasons for that might be varied, but the one thing that we know is that we are finding fossils, especially now that paleontologists have a very good idea of where to look, because our dating mechanisms or our dating strategies for finding where we ought to go in time have improved so dramatically. We use radiometric dating, and we are able to look in the right place rather than probably just look everywhere. Could you hit the space bar for me? So what I'd like to spend some time on today is talk about what I would call the new evidence for evolution. This is the kind of evidence that would absolutely blow Charles Darwin's mind. I mean, this would be so amazing for him, because you have to remember, at the time that Darwin came up with his theory, he had a very unsophisticated notion of heredity, and certainly a genetic mechanism had not been worked out. For Darwin, genetics was more like traits come from blending. It's like making different colored paints, right? You take paint of this color and that color and you blend it, and then you get an offspring that has a particular trait. Well, as it turns out, that's really not the mechanism at all, and we know now that heritable units are discrete traits, they're genes, and we know now, based on some of our recent advances in the fields of microbiology, and particularly the field of genomics. And so genomics is a field where we can take an individual organism and we can read the entire set of instructions. Everything about that organism is in that code. Now, without supercomputers, that certainly would not have been possible. But there's an amazing phenomenon that's going on today, and that is that the ability to sequence a complete genome is rising at an exponential rate. So today, we can sequence about 9 billion base pairs in a single day. 10 years ago, that number was 1,000-fold lower. And it's thought that within a very short amount of time, we'll all be able to get our own complete genome sequenced for under $1,000. So the price of that's going down. Now, there are certainly some practical reasons why that could be beneficial to us, and as we'll see, it turns out that understanding genetics is not quite as simple as Mendel and the pea experiments that he did. Next one, please. So this is just a plea to you that if things get sort of carried away during social hour, if someone could make sure I get home safe and sound, I would appreciate that. So what did we learn? And again, Mendel did publish his work while Darwin was still alive, but the interesting thing is it wasn't really touted at the time, and went into a period of obscurity for a number of years until other individuals were doing similar sorts of genetic studies. And is often the case in science, when you're working on something and you go to the library, it's bitterly disappointing because you find out that someone has already had your good idea. It's not a fun moment. So at any rate, we'll talk a little bit about some of the basics of Mendelian genetics and some of the things that move past that. And we'll talk about this idea of what I'd like to think of as the code for life. And we're fairly familiar with the concept of genes, but as it turns out, some of the interesting things that we're learning about the relationships among organisms has to do with the genes, but also with this other assemblage of DNA that we used to call junk DNA in sort of a dismissive fashion. And I think we've become more sophisticated to know now that there are a lot of features about DNA that we're still learning about. The whole mechanism of folding and winding is very complicated. So it may turn out that things that are non-coding and used to be called junk actually do have some importance. But they have an interesting property that I'll get to in a little bit that really gives us some great information in terms of looking back historically in evolutionary time so that we know something about how we're related to each other and how we're related to other living organisms. And part of this is done by not looking at the typical things that people talk about in evolution. We talk about mutations that lead to adaptations and various traits and at some point speciation. Well, all of that is certainly important, but there are also these neutral mutations. And the neutral mutations, which can occur in this non-coding DNA, don't lead to adaptations. They're not selected for, they're not selected against. But they leave a trail, they leave a trace and a history in terms of these mutations occurring. Now, the way that genes come into existence is that they aren't a result of many mutations and voila, you have a new gene. Rather, the typical fashion is that an existing gene, through some minor error in replication, duplicates itself. And that duplicated gene might then accumulate some mutations that could be useful. If so, it's activated. If not, it may end up not being a gene that we use to make proteins, and in other words, would be called a pseudogene. So these neutral mutations and pseudogenes give us a really good indication of how we can look at the relatedness of organisms. So simply here then, I mean, this is the code. If my computer, which can do some pretty sophisticated things, uses a language based on a pretty simple binary code, zeros and ones. So with something that simple, I can get some pretty amazing programs to run on my computer. Well, we're based on a simple code as well. And our code is just A, T, and C, and G. And our complete set of instructions are encoded on those molecules in these very, very long linear polymers. And these linear polymers were a huge mystery for a long time for biologists, because it would be like having a string of numbers of 0, 0, 1, 1, 0, 1, 0, 1, and being even if you could read it, what would it mean? And how do you find where you are in the code? Because it's just so repetitive. Well, that was a huge problem for geneticists, and a huge problem in terms of trying to understand, once we realized that DNA was actually the molecule that encoded for heritable traits, how would we actually access it and read it? So the other thing you probably know about DNA is that it's this interesting double spiral molecule. It's a double helix. It has what's called complementary base pairing. So we always see G's on one strand bound to C's on the other. And it has to do with the chemical interactions, the weak hydrogen bonds that form, that hold it together. It's sort of like having two different facets on Velcro that only bind with each other on opposite strips, and then taking these Velcro strips and twisting them up and making nice, pretty shapes out of them. If you want to, you can apply a little bit of force and pull that Velcro apart, just as you can break these hydrogen bonds, which is required to do if you're going to replicate this to make new cells, or if you're going to make a copy of the DNA into another molecule, an RNA molecule, that then goes and makes sort of the business end of organisms, which are the proteins that make our hair and our skin, and the enzymes that digest our food and detoxify ethanol, those sorts of things. Next. All right, so how do we read it when we're interested in reading it and learning something about the history of life? You know, where we came from, who are these other guys, and how might we be related? Well, so the first thing we had to do is basically decipher this code. And that's a really interesting story in and of itself. I don't have a lot of time to go into it, but as it turns out, we basically figured out this is ATCG really form 3-letter words.